Hello everyone, my name is Shraddha and welcome to my YouTube channel Node of Biology. In the previous video, we started discussing about the common structures found in all types of cells and we discussed one of them in detail, the plasma membrane. In case if you haven't watched that video, please follow the link in the description box. Today, we will be discussing another two structures found in all types of cells, the cytoplasm and the ribosomes. Now let's discuss the first structure, the cytoplasm. The cytoplasm is the fluid-filled interior of a cell found in both prokaryotic and eukaryotic cells. In case of eukaryotic cells, the cytoplasm includes the entire content within the cell membrane except the content of the nucleus. Since the cytoplasm is packed with different molecules and cellular components, most of the chemical reactions of a cell occurs in the cytoplasm, including glycolysis, protein synthesis, and other metabolic reactions. The cytoplasm is made out of three key elements: the cytosol, cytoplasmic organelles, and inclusions. Cytosol is the fluid-filled part of the cytoplasm, which is mostly made out of water, some dissolved ions, and water-soluble molecules. The cytosol does not hold any of the organelles. Cytoplasmic organelles in the form of mitochondria, Golgi apparatus, endoplasmic reticulum, vacuoles, and others are found in the cytoplasm, and inclusions in the form of carbohydrates, lipids, amino acids, enzymes, and other molecular compounds are also found in the cytoplasm. It is because of the cytoplasm that the cell has a shape, and this cytoplasm also helps the different components to navigate around the cell simultaneously to undergo different chemical reactions. Some key functions of cytoplasm include: it provides shape to the cell, it helps in the movement of molecules and cellular components within the cell, it is a site for synthesis of protein, it is a site for production of ATP via glycolysis. Different molecules such as amino acids, carbohydrates, and others are dissolved in the cytoplasm, and cytoplasm also helps in dissolving certain waste products. A quick summary of cytoplasm would include the following points. Cytoplasm is the fluid-filled portion of the cell. Structurally, it is made up of cytosol, the cytoplasmic organelles, and the inclusions. Cytosol is the fluid part of the cytoplasm, which is made up of water, dissolved ions, and other soluble compounds. The cytoplasmic organelles in a cell includes the mitochondria, endoplasmic reticulum, Golgi apparatus, and others. The inclusions include carbohydrates, amino acids, lipids, enzymes, etc. The location of the cytoplasm would be the entire content of the cell within the cell membrane. In case of eukaryotes, it will exclude the nuclear contents. Functionally, the cytoplasm is involved in most of the chemical reactions, such as glycolysis, which does ATP production, or protein synthesis by ribosomes, and other metabolic reactions. Now let's discuss the next common structure found in all types of cells, ribosomes. The ribosomes are also known as ribonucleoprotein particles. They are particles and not organelles because they do not have any membrane. They are found in abundance in all types of living cells, and they serve as a site of protein synthesis. Structurally, the ribosomes are made up of proteins and RNA. The RNA in the ribosomes are known as ribosomal RNA. Each ribosomal particle is made up of two subunits, a large subunit and a small subunit, and individually each of these subunits are made up of RNA protein complexes. On the right side is an illustration of the structure of a ribosome undergoing translation of an mRNA molecule. In the illustration, the orange colored unit is the large subunit of the ribosome, and the white colored unit below the mRNA molecule is the small subunit of the ribosome. The green colored structure is the transfer RNA, or labeled as the tRNA. This tRNA brings amino acid to the ribosome. They specifically act as a bridge which matches the information hidden in an mRNA molecule with the amino acid it is supposed to code for. Thus, the ribosomes are involved in organizing the process of translation, and catalyzing the reaction which joins the amino acid to make a polypeptide chain. Now, when a cell is about to undergo protein synthesis, these two subunits unite around an mRNA molecule and form a complete ribosome. Once the structure is complete, the ribosome starts to move forward on the mRNA molecule. Then it starts reading the information hidden in the mRNA molecule and starts translating this information into a polypeptide chain. Once this process of translation is finished, these two subunits are pulled apart so that they can be reused again when the cell plans to undergo protein synthesis. Location: in prokaryotes, ribosomes are freely floating in the cytoplasm of the cell. In eukaryotes, they are free or attached. They are attached to the endoplasmic reticulum. Functionally, they synthesize proteins to perform cellular functions. The area of protein synthesis in the cell determines what type of protein it can make. For instance, free ribosomes usually make proteins which function within the cell. For example, they make structural protein. Whereas attached ribosomes or bound ribosomes, they produce proteins which can function either inside or outside of the cell. For example, signaling proteins. Now, why does a cell need to make proteins? Why does the ribosomal subunits unite and read the mRNA molecules to synthesize a growing protein chain? Proteins are needed to speed up the biological processes in an organism. They build structures within the cell, for example, cytoskeleton. They control the production of other proteins by controlling their synthesis. They transport different molecules across the cell membrane, and they can speed up the chemical reactions using different enzymes. Now let's discuss the similarities and differences between prokaryotic ribosome and eukaryotic ribosome. The similarities include that ribosomes in either prokaryotes or eukaryotes are made up of similar structure. That is, they are composed of a large subunit and a small subunit which unites when a cell undergoes or plans to undergo protein synthesis. Once they unite, they form a complete ribosome. Then they start reading the mRNA and start producing the polypeptide chain. Functionally, ribosomes from a prokaryote or eukaryotes both undergo protein synthesis. The differences include that in case of prokaryotic ribosomes, the instructions to make proteins are obtained from the cytoplasm, whereas in eukaryotic ribosomes, these instructions are obtained from the nucleus. The prokaryotic ribosomes are smaller than the eukaryotic ribosomes. Prokaryotic ribosomes are known as the 70S ribosomes, which are made up of 50S and 30S large and small subunits. The eukaryotic ribosomes are known as 80S ribosomes and are made up of 60S and 40S large and small subunits. In case of prokaryotic ribosomes, the RNA content is much higher than the protein, whereas in eukaryotic ribosomes, it is opposite. The protein content is much higher than the ribosomal RNA content. The diameter of a prokaryotic ribosomal particle is around 200 angstrom in comparison to a eukaryotic ribosome particle, which is around 250 to 300 angstrom. A quick recap on ribosome would include the following points. Ribosomes are ribonucleoprotein particles found in abundance in all types of cells. Structurally, they are made up of RNA, ribosomal RNA to be precise, and proteins. Each ribosomal particle is made up of two subunits, the small and the large subunits. In case of prokaryotes, it is known as the 70S ribosomes, made up of 50S and 30S subunits. And in case of eukaryotes, they are known as 80S ribosomes, made up of 60S and 40S subunits. These subunits assemble when the cell is about to undergo protein synthesis and are pulled apart once the process of protein synthesis is over. That is, DNA is replicated into another DNA molecule, which is then transcribed into an RNA. This RNA is then translated into proteins. The location of ribosomes could be either free or attached, and their chief function is to undergo protein synthesis. For future videos, please like, share, and subscribe to my channel, Node of Biology.